So thank you for inviting me today. I'm going to talk about the genetics of intracranial aneurysms. And so first I would like to disclose that I have nothing to disclose. And IA is a very complex phenotype caused by genes, environmental factors, and their interactions. We know some of these risk environmental factors, such as smoking, a very important one. And then we do know now some susceptibility loci. I'm going to talk about their identification today. But these interactions will be critical, so that will be kind of the bulk of the common complex forms of intracranial aneurysms. And then it's worth noting that about 5% of all of these cases are associated with inherited connective tissue disorders. So it's very important to ask about the family history of these connective tissue disorders. And so this is just giving you an overview about the similarities and differences in the different types of aneurysms. And now if you look at I'm going to focus on the IA there, you can see that the prevalence is about 2%. And then if we think about the genetics there, so we can see that there is clearly an increased risk for relatives of siblings of these patients. So we are talking about relative risk between 2.5 and 7. And also if we look at the prevalence of among first-degree relatives of these patients, it's 10. So these are high risks. And so what are the risk factors for rupture of IA? So this was one study published a couple of years ago in stroke, and that was a large study, and they identified that these are the important risk factors. These are kind of the key things that, of course, when we talk about IA, that we are interested, what are the risk factors for rupture? And that's also something that we would like to think about in terms of the genetics, so this is the kind of the main focus. And now how can we identify genes for complex traits such as intracranial aneurysms? So typically what we do nowadays is that we do a genome-wide association analysis. So in that analysis, we will screen several thousands of variants throughout the genome and test those variants for association with the trait. And then we can get significant peaks given the multiple testing. So we have to take into account the multiple testing there. And there, after that genome-wide analysis screen, so then we do a replication study. So the key peaks, the key markers there are then we try to replicate those in an independent study sample. And after that, we can do regional analysis, extensive sequencing of the region, and look for the linkage disequilibrium structure among the variants, and then finally do some functional studies. And so what are the typical study samples for complex traits, genetic studies of complex traits such as IA? So we have these are the most typical ones used. So we have cases and controls, or we can select families. So we can perform family-based association studies. And there we typically collect at least the parents and the proband. But then in the family-based association studies, there often is the difficulty to get both parents, and also it would be perhaps more a genetic approach to start with families where there are multiple affected individuals. So you can see that it easily leads to kind of much more complicated collection process than collecting just cases and controls. And then, when we think about the case control studies, that is now the main form to do genome-wide association studies. it may be difficult to establish that when the association arises, that what is the direct causality between the factors that we are investigating. And it's critical, totally crucial to think about the distribution of age, sex, and ethnicity between the cases and controls, because those issues can cause stratification bias. So that's very important to focus on this. And in the family-based association, they are kind of a safe method in the sense that they have been developed exactly to circumvent this problem of related or relevant controls, especially if we use mixed populations, such as a study sample from the Los Angeles area. And so there in this setting, so the internal control is then derived from those alleles that are not transmitted to the affected individuals. And so I said that we are using this genome-wide association. So the reasoning why these were studied for complex traits was that there were papers showing that these association tests are much more powerful statistically than the typical linkage analysis when we try to detect alleles of modest effects. And also we will perform these using singular nucleotide polymorphisms because the genotyping platforms for these type of markers are much easier and more cost efficient than the ones for the microsatellite markers. So these are different types of variants that we have throughout the genome. And so this is also an unbiased way regarding what is the function of the variant and where is it located. So we can kind of detect all different types of variants. And also, as I said, so this has been made possible by recent genomic advances. So the technology is now allowing us to genotype in one array up to even more than 1 million markers at the same time. And so this is one of the examples of a meta-analysis of genome-wide association studies for lipids. So we have different traits here. We have HDL cholesterol, LDL cholesterol, and triglycerides. And then this genome is screened throughout the chromosomes. And we will have some interesting peaks here. So they are implicated, and they show here in this particular study, they show us which ones are the previous lipid genes that peak. But they also have some peaks that are totally new peaks for lipids and that have never been implicated for lipid traits. So that's the beauty of the genome-wide association studies, that you can also identify totally novel genes that were not connected to these traits previously. And this is a key region that was identified for coronary heart disease. Here we have the risk interval. It's about 58 kilobases long, and we have several associated variants here indicated by these arrows, and then they are in tight linkage disequilibrium, so they kind of are redundant markers. And then if we look at the structure there, we have a couple of genes there, and then we have so here is again the risk interval, so but nothing really like here, so the signal is between the genes. And this region has been a very strong signal for coronary heart disease. The mechanisms are not fully clear as of yet. And interestingly, the same region is also associated for IA. And so this was the first genome-wide association study published for IA. And that was performed a couple of years ago using 3 different cohorts. The first one was a Japanese, then a Dutch and Finnish. And the total study sample was about 12,000 individuals. And they identified 3 significant Lodge scores, peaks. And if you look at the odds ratios, they were relatively low. These are typical odds ratios for genome-wide association studies. So these variants had a minor to modest risk. And I'm coming back to these particular genes that they identified a little bit later, but basically, this year there was the second GWAS study for the trait, and here they expanded the sample size to increase their power. So now in this study they had a 89% power to get relatively low risk variance, so genotypic relative risk of 1.25. And if you look at now the number of cases, so they have nearly 6,000 cases in this one. And they added 8 new cohorts and expanded one of the previous cohorts here. And what they obtained there was that they were able to replicate or confirm 2 of the previously found loci, and they identified 3 novel loci. And now you can see again that the chromosome 9 loci that has been implicated for coronary heart disease, that was also again one of the loci that was strongly showing up in this scan. And so this chromosome 8 locus was actually the only one that didn't quite reach the genome-wide significance, and the replication was not there for the replication study sample. And again, if you look at the odds ratios, they are very low, but typical for genome-wide association studies with common variants. So this is the region on chromosome 9p. You can see here the highly associated marker, and also you can see here that if they look at the different study samples, it's pretty consistent in terms of the odds ratios, and then these are the overall results. So you have an odds ratio of 1.32. These genes that land in the region, there are now a lot of investigation around this involving the function of these SNPs. So they are considered tumor suppressors and cell growth regulators. And both genes have been also found to be deleted in many cancer cell lines. And as I said, so the same region is associated with coronary heart disease. So now the question is that what is the underlying mechanism, how this region is causing this signal for both coronary heart disease and aneurysms. And the sequencing is kind of the next step that people are sequencing and also making animal models involving the regional genes. And so the conclusions of the second GWAS were that so many of these genes are implicated in the or have a role in cell cycle progression, possibly influencing the proliferation and senescence of the progenitor cells responsible for vascular formation and repair. So they have a very interesting function in terms of these phenotypes. But now if you look at the risks or how much these variants actually explain, so you can see that these 5 GWAS loci account for a very small percentage of the familial risk of intracranial aneurysm. So the Finnish cohort had the highest explanation percentage, but still this is just a minor part of the familial forms. So you can see that now screening the common variants, this is the end point. This is how much we are able to explain. So that indicates that that there are a lot to do in terms of genetics. So we have to find the rare and low frequency variants that were not screened. So we were kind of optimistic thinking that these common variants would explain more about the phenotype than they did. And this is a very common phenomenon. This has been identified. The same thing has been identified for diabetes, asthma, for many of these complex traits that are caused by multiple genes and environmental factors and their interactions, that the GWAS signals actually explain a very small part of the variance of the trait. And so there was also a very recent paper that I saw in Stroke showing that smoking actually greatly increases the risks of IA in patients with variants on the chromosome 8 and also on 9. So that's interesting. I think that to really explore how much these variants explain, so we also have to take into account the environmental factors and their interactions with these variants. So this is a good study showing that. What are now our ways to identify novel genes for aneurysms? So the thing that's already going on is that people are following up and doing so-called targeted sequencing of the 5 GWAS regions on these human chromosomes that were implicated in the second GWAS study. And then another way to look into is to do exome sequencing of extreme IA cases, and those cases should then also have family history to really emphasize the familial component. And then we can also do something called RNA sequencing of tissue samples derived from the IA cases and controls. And we have actually started a collaboration, as Dr. Gonzalez mentioned, so with Drs. Vinuela, Gonzalez, and Bui here at UCLA. And our aim is to collect a large IA case control study sample and then utilize these novel approaches. So most likely we won't be following up so much the already identified regions, but definitely we have interest in exome sequencing of extreme cases such as the one presented today with clear family history. And then also we are interested in methods looking into the gene expression in the tissues or blood samples of these individuals. And so as I said, the genome-wide association studies, the results of which I showed, so 2 have been published. So those typically identify common DNA sequence variants with minor allele frequencies more than 5 to 10%. So they are very common variants that typically explain little in terms of the odds ratios. And then we have the extensive resequencing, and that is needed for the rare variants, so variants that have minor allele frequencies less than 1%, and also low frequency variants that were missed in the genome-wide screens. So now the recent technological advances, the next generation sequencing methods actually enable these extensive targeted resequencing. And how people are looking into this resequencing projects are that we are taking the population extremes of the trait of interest, or that can also be the extreme cases and very healthy controls, and very carefully screened controls so that we really know of the controls that they are healthy. And then the extended families are a very advantage study sample as well, because there we already know that there is a likely genetic predisposition. So people are focusing on the extremes when they look for the rare and low frequency variants. And then, so here the first procedure is to do sequence capture. Of course, the aim is to sequence the entire genome in future, and that's already done in kind of a smaller scale. So genome sequencing is kind of more and more genomes are being sequenced, but still it's relatively expensive still to do, though it's becoming cheaper every month. But typically we still start with the sequence capture and then do the massive parallel signature sequencing and follow up then the variants in extensive case control study samples or in these families. And so this is just a basic idea when we do sequence capture. Let's say we would be interested in this chromosome 9. So we would first take the individual's DNA, shear and ligate the adapters. Then we would mix the DNA with the biotinylated RNA. So that's the key step there. And then finally we would be able to use the magnet to selectively pick up the beads, allowing then also the unbound DNA to be washed away. So now we have there the biotinylated RNA and the complementary DNA complexes bound to the beads. And then the captured DNA is then PCR amplified using just universal primers, and we are then able to run the DNA on the sequencer. And now we have the targeted region. We only sequence the part that we are interested in. And the similar idea can be utilized for exomes, so we can take the entire coding sequence of a person and just sequence the coding. And so that's the exome sequencing. It's called targeted exome capture. So now we are just focusing on the coding sequence of a human being. And this is a very effective strategy to just selectively sequence the coding. And I would like to do that in extreme familial IA cases that are very likely to represent monogenic forms of IA. So that would suit perfectly for that. And then also, as I said, the whole genome sequencing is not quite yet feasible, mainly due to the high cost. The technique is there, so it's the cost. And then so the exome, now when we talk about exome, so that means that we are interested in the coding, so all exomes will be sequenced, and that requires about 5% as much sequencing as the whole genome. So we are talking about way less sequencing here. And the coding constitutes only about 1% of the human genome, so we are sequencing, in fact, about 30 megabases. And now protein coding regions contain a large percentage of the disease-causing mutations, so therefore it's of interest to focus on this sequence, although recognizing that we are missing intergenic and intronic pieces of the genome. And so this is just the idea there, that what do you do? So you have these extreme cases, and you are looking for certain types of coding variants, and then you also have to have control exome sequence. So it's critical to compare your sequencing results to these control exomes, and then also utilize the public genetic databases when screening for the important variants. And then you are using these two filters, you are able then to exclude common variants, and then you are left with your candidate genes in the end. So you kind of screen off the already known common variants that are present in the genetic databases and in the exome sequencing controls. And so typically the reason why only less than half of the 7,000 rare monogenic disorders have been discovered is that the identification of these genetic variants for rare disorders has been limited by just the sample size of the affected individuals, reduced penetrance, locus heterogeneity, and alleles that impair reproductive fitness, so we may have very small family sizes. So it's been very difficult to get large enough study samples to investigate the rare forms of the common disorders. And now that has been changed by this exome sequencing. So it's really heavily now used to look for these remaining monogenic disease genes. And this is just an example how they actually did it in the very first, I think this is the second or third exome sequencing paper. There they utilized these filters. They used the common databases for the variants, and they looked for certain types of variants, and then they utilized the control exome sequences, and then they finally left with a very large amount of variants and ended up with just one variant, or one gene, sorry. So that was a very clever way how they got rid of kind of the noise in the system using these filters, and then ended up just with one gene and identified actually a gene for a rare Mendelian disorder using this technique. And these papers changed kind of the way how we are now looking at the rare disorders. So we will be and are using now heavily exome sequencing to identify genes for rare disorders. And a couple of considerations that we have there is that these causal variants should be unlikely then, of course, to be found in these public SNP databases, so single nucleotide polymorphism databases, or in the control exomes that will add the noise if that's the case. And then the genes for recessive diseases or traits will be easier to find because fewer genes in any single individual have 2 or more new or rare non-synonymous variants. And then genetic heterogeneity, of course, will hamper this analysis, so it would be beneficial to have the same ethnicity for the study sample, and that's an important factor, or utilize individuals originating from a genetic isolate, such as, for example, Finland. And limitations when we do exome sequencing is that there may be some missed variant calls, we may have low coverage, or some variants are not identified easily. And also the filtering, it relies on the public databases, and they have uneven ascertainment of variation across the genome. And then we also need catalogs, so we need this control exome. So that's very expensive to do, so to sequence exomes. So now the 1,000 Genomes project will, I'm coming back to that, but that will provide a catalog soon about the estimated allele frequencies. And it may be also necessary to establish frequency-based thresholds for what is kind of a common variation that is unlikely to be causal for disease. So all of these limitations need to be thought of when we think about exome sequencing in AI. And so this is the project I was talking about. So this is now currently building the most detailed map of the human genetic variation to date. It's supported broadly. And they just released data from 3 pilot projects this June. In the first pilot, they sequenced the genomes of 6 people. So these were 2 nuclear families, each with 2 parents and a daughter at a very high coverage. And then they sequenced the genomes of a little bit less than 200 people at low coverage, and also they sequenced exomes or actually not exomes, but exons of 1,000 genes in 700 people. And this data is now publicly available, so that's the key advantage and benefit of this project, that it is providing the data freely accessible, so you can use their or download it from their websites, and then centers that don't have the capacity for the enormous amount of data that's there, so they can use the computer clouds available in web services. And then another benefit is that they are now sequencing genomes of 2,500 people from 27 populations around the world. So that will be very beneficial data for these type of studies as ours, that we will need controls. We need the control exomes, and we need the control sequences. And this data can be used for our studies and to follow up the GWAS signal. So it will be really a very, very wonderful resource for all people working with genetics of complex traits. And so the last thing I wanted to mention is the transcriptome analysis, so the RNA sequencing. And this is something we are very interested in our project, so that we would like to take a blood sample and then isolate the RNA, lymphocyte RNA, and then look into the RNA and the expression of the genes in the cases versus the controls. And when we think about that, when we have RNA of these individuals, so it can be arrayed using the traditional microarray methods. But when we do the sequencing of the RNA, that's really beneficial, because then you not only get the gene expression at some points of the gene, but you get the gene expression levels throughout the entire gene, because you are sequencing, not just looking certain parts of the genes as in microarray methods. And also you can get the all the expressed coding variants, and you can characterize known and novel splice forms, and that's something that's never been investigated for aneurysms, that are the splice forms that are more important than other. And also we can look for allele-specific expression of these variants, whether a variant actually changes the expression in an allele-specific way. So that's very interesting, and something that's now becoming available. And then we can do a joint analysis of these data for the IA phenotypes, also utilizing some network approaches. So that's kind of the where we are heading now with the blood samples from our study sample. And finally, I would just like to acknowledge the people who've been involved in this project. So we are just in the beginning, and hopefully we'll get exciting novel findings for aneurysms. Thank you.